Let's take a quick check back to the specification then and see where we are. So hopefully you're happy with the fact that we can investigate genetic diversity by looking at measurable, or observable characteristics. So looking at these things that we can directly measure, or we can easily look at the DNA-based sequence. What we're going to think about now is why is the mRNA-based sequence also a useful way and a useful sort of measure that can tell us about the diversity and relationships between organisms. So as well as looking at the DNA-based sequence, we could look at the mRNA sequence and look at the nucleotide sequence in that mRNA. And the reason we can do that is we know that our base sequence on our mRNA strand is directly complementary to the DNA-based sequence it was transcribed from. So mRNA gives us a really good representation of what the DNA-based sequence was. And so we can get, just as sort of as good a indication of the genetic diversity by looking at this mRNA sequence. The only problem is that, and you should be able to think about this from the work we did on protein synthesis, mRNA sequence doesn't give us as full a picture of the DNA-based sequence. Now, just take a second to think about why that could be. Now, hopefully, if you're thinking about how mRNA is made from the DNA-based sequence, hopefully you've recognised that in the process of transcription, and before that mRNA strand leaves through the nuclear pores to be translated into an amino acids sequence in a polypeptide chain, we get this process of splicing. Okay. Now, if you remember, when you first transcribe your DNA strand, that DNA strand forms pre-mRNA, but within that pre-mRNA, it will contain both the exons and the introns. Now remember, we want to only have in our mRNA strand the coding portion that's actually going to code for amino acids. And so when splicing occurs, obviously that removes that non-coding DNA section, which is fine, and that's great for protein synthesis. It means that we don't transcribe unnecessarily, unnecessary information that we don't necessarily need to make the protein. But if we take out those non-coding sections, then that means that we don't get as full a picture of the DNA-based sequence. And what we've got to remember in evolutionary terms, it's not just the coding parts that are important for us to look at when we're looking at how closely related organisms are. Okay, so quick tick back and syllabus check again. So we're good for the characteristics, we're good for sequence of DNA, and hopefully now, really simply, we should be able to kind of link in how we can use the sequence of mRNA and the limitations of using that mRNA sequence due to the whole issue of exons and introns and splicing. So let's now have a look at how we might be able to use the amino acid sequence of the proteins encoded by the DNA and the mRNA to investigate diversity. So in this instance, what we can do is we can take the same protein from different species and actually compare the sequence of amino acids in that protein to determine how closely related or distantly related those organisms are to one another. And that's possible because of that whole relationship between DNA, let's make my A look a bit more like an A, mRNA, and the fact that that then codes for the sequence of amino acids in a polypeptide chain. Okay, so obviously if we look at the amino acids, that can then tell us about what the mRNA sequence was, and when we know the mRNA sequence, that can tell us about the DNA sequence. And actually, in terms of sort of practicalities, sometimes it is easier just to look at the sequence of amino acids rather than do all that DNA kind of analysis and try and get the full DNA-based sequence. So it makes it a really good way of looking at how closely or distantly related organisms are to one another. So we'll have a look at some actual examples. So let's have a look at what information we've got here. Here we've got samples of the same protein And that protein, I mean, you haven't got the whole protein here, but a section of that protein made of 7 amino acids long. So we can see here are the 7 amino acids long in this particular protein from 6 different species. Okay, and what we're going to do is look at the similarities and the differences comparing different species with each other. So for example, and I'm going to write the similarities in blue in this box here. So we're going to call this side similarities. Okay. And so if we compare, obviously we can't compare species 1 with species 1, and you can't compare 1 and 1, hence why this bit here has been scribbled out. You can't compare 2 with 2, so this bit's been scribbled out. So what we'll do first of all is we're going to compare species 1 with species 2. So I'm going to put information into this box here. So if I have a look, working along, comparing species 1 and 2, looking for how many similarities in their amino acids, well, I can see that they've both got Phi in that position, so that's one similarity. Met and Ala, so that's a difference, but then they've both got Arg, so that's a second similarity. They've both got Sur here, so that's a third similarity. Both got Glue, fourth similarity. Difference here, Val and Met, okay, but they've both got Ala at the end, so that's a fifth similarity. So between 1 and 2, number of similarities is 5, and if you just change colour of pen, between 1 and 2, number of differences, well, we know that Met and Ala were different, and Val and Met were different there, so number of differences is 2. Okay, what I'd like you to do is have a go at completing the rest of the table, show your similarities on one side in the blue, and then add your differences. Okay, so your differences on this side in black. Okay, give you some chance to have a go at that, and then when you think you've completed it, switch it back on and double check your answer against the completed table. Okay, so let's see how you've got on. So when we compare 1 with 3, you should have found 5. 1 with 4, there were 6 similarities. 1 with 5, there are 5, and 1 with 6, there are 6. Then if we take a comparison of 2 with 3, you should have found 7. When we do 2 with 4, you should have found 4. 2 with 5 is 3, and 2 with 6 is 4. Then when we do 3 and 4, you should have found 4. 3 and 5 is 3, and 3 and 6 is 4. Then if we take 4 and 5 together, you should have got 6. And then 4 and 6 together, you should have got 5. And then our final little one, 5 and 6 here, there would have been 5 similarities. Okay, let's do our differences. Change of pen. So in terms of differences between 1 and 3, you should have found 2 differences. 1 and 4 is 1 difference. 1 and 5 is 2 differences, and 1 and 6 is 1. Between 2 and 3, there were 0 differences. Between 2 and 4, there were 3. Between 2 and 5, we should have found 4. 2 and 6 is 3. And then working down, 3 and 4 is 3. 3 and 5 is 4. Then we've got 3 and 6 is 3. Then 4 and 5 is 1. 4 and 6 is 2. And then finally 5 and 6 should be 2. So that should be our completed table showing the similarities and differences between that protein that we're thinking is kind of the same protein in these 6 different species, but we can see even though it's the same protein, we can actually get slight differences in the amino acid sequence, And we know if the amino acid that's been put in that order of the polypeptide chains is different, it must mean that the mRNA-based sequence was different, because that would have provided a different triplet for that particular amino acid, which means our DNA-based sequence must have been different. So what you can now do, based on the information you've got here, is make some decisions about what you think is most closely or more distantly related. So if we take a look at species 2 and 3, so we're having a look at species 2 and 3 here. If we have a look, they had 7 similarities in common. Okay, so they literally only had, well, they didn't have any differences. So what that might bring you to suggest, but the evidence based on from just that protein, is that those two organisms are very, very closely related. If we take species 1 and 4, they in their amino acid chain for that particular protein had only one difference. So that might suggest that they were more closely related to. So 1 and 4 would be more closely related than perhaps if we compared, the ones where we have more differences. Now, just based on this table, actually, quite a lot of these sequences were quite similar. But if you have a look, maybe comparing species 2 with species 5, to have 4 differences, that would mean that they were fairly different. Now, to make things a bit more complicated, a bit more exciting, what you can be asked to do when you're applying your knowledge of this is to construct a phylogenetic tree using the information about the number of amino acids that they've either got in common or different. So in this table here, we've got, they've taken samples of hemoglobin, which we know is a protein, from these different species here. So from a human, a gorilla, a gibbon, a rhesus monkey, a horse, a kangaroo, a chicken, a frog, and a sea slug. Now, what you should be thinking to yourself is, well, yeah, I can just recognise there that I'm probably going to be more closely related to a gorilla and a gibbon than I am to a sea slug. Well, you can use the evidence just by looking at the number of amino acids that are different. So in this situation, they've used the differences from human hemoglobin. So obviously, human hemoglobin is going to have no differences from human hemoglobin. Gorilla hemoglobin has just got one difference, whereas if we look at chicken hemoglobin, there are 45 differences. Sea slugs, 127 differences. So what you would do is try to construct a phylogenetic tree using the information in this table, grouping together those that seemed like there were very few differences quite closely together, because they would have shared a common ancestor more recently, whereas those with the bigger differences more further apart. So hopefully you'd come up with something like this. Okay, so here's us as the humans, and we can see, based on the information we were given, there was only one difference in our hemoglobin, so we'd suggest that we shared a common ancestor really quite recently in evolutionary history, whereas the sea slug, where we had many, many differences in our hemoglobin and the amino acids in that hemoglobin, we must have shared a common ancestor a long way back in evolutionary history. So we can just use these simple patterns to put together kind of an estimation of those relationships.